Great. Thank you, everyone, for attending, and I appreciate for the organizers for allowing us to talk. This is a great opportunity and a fantastic crowd. So I presented this morning on the ClinGen data access as it related to APIs and streaming services, and I mentioned the Allele Registry and that. And so I'm going to touch upon the introductory materials a little bit. So if you attended that, it'll be a little bit of similarity, but I'm actually going to be talking about a lot of the other functionalities of the Allele Registry, mostly from the user interface perspective. Okay, so first off, I'd like to go over the goals I'd like to cover for today. First off, I'd like to talk about the Allele Registry solution for canonical variant naming and its purpose and what services use it. The external sources that we ingest that populate and seed the Allele Registry, aside from the users that also import their own variants for naming services purposes. I'm going to briefly go over the community usage statistics, briefly mention the Allele Registry API, and I'd like to do a live demo. I'm going to use a buyer curator use case for explaining some of the additional tools that we have in Allele Registry besides the query interface. I'd like to talk about a little bit of structural variant support in Allele Registry, and depending how much time we have, perhaps a live demo of what some of the queries are in the Allele Registry. And I'd like to leave some ample time for questions. Okay, so the proposal of the ClinGen Allele Registry is essentially attempting to canonicalize variants. And what do we mean by that? At the top, you'll see this diagram here. We start off with a genomic canonical allele. I'm going to show some additional examples of this in a little bit. But first, you'll see here at the bottom, you see these genomic alleles. And essentially what this is these are different genome assemblies that we have in Allele Registry, namely GRCH38, GRCH37, and NCBI36. And you see these contig accession names. So this is chromosome 11, and we have a genomic coordinate, and we have a reference allele, and we have an observed allele. So the reference allele in GRCH38, and have to be the same for the other ones, is C, and then the observation is this T. And so what this attempts to do, the service, is actually contextualize these alleles, and that we actually can canonicalize or have a variant naming that goes across these different transcripts and genomic coordinates that represent essentially the same effect and the same coordinates. And what do we mean by same effect? That means the same alignment across these different entities. And so this is originally described in this 2018 paper here in published in Human Mutation. And so what the solution attempts to do is supporting the stable and consistent variant identification and naming, namely for curation and pathogenicity. And this is one of the FDA requirements for classification processes. And what you see here is the CAIDs. And so this represents canonical allele identifier. And this is one example of CA321211 is one such identifier. And what we try to do with the system is provide for open sharing of genetic variant information and aggregation of this information from different sources, along with the links to those sources. And we attempt to abide by these FAIR principles. Essentially, it's being able to find the data, access the data, make sure the data is interoperable between tools and different uses, and it's also reusable. And so you can actually reuse some of these services that we've exposed to the APIs. We've also wanted to provide these identifiers on demand at high bandwidth and low latency. And this is very important for these very large scale data sets that produces millions or hundreds of millions of variants that allow us to get up into the billions of registrations in the Allele Registry. So we also provide API support for programmatic registration and access, which supports large batch queries and registrations, and also provide this JSON output, which is a JavaScript object notation, which is a common structured notation that's modeled to allow people to obtain the data and actually be able to basically take out certain parts of the data of interest. And we also we're sure to make sure that this is free and open user registration. So it's on demand. So as soon as you sign up, you're able to register your variant right away. And so we have this URL here at the bottom. It's reg.clinicalgenome.org. And I'm going to be showing some live versions of that in a little bit. Okay, so we talked about an observed variant. So this would come from a variety of sources. So oftentimes people might see an observation or something that says a ClinVar variation ID. So that's a named variant, and you can go to this and you can actually see what also is available for this. You can see there's a lot of information here. If people are familiar with ClinVar, it's an excellent resource. And it's one of the many sources that that we ingest into the Allele Registry. And you also have these other contexts when people actually would like to register and name a variant. And oftentimes you'll either start with a chromosome build, GRCH38 in this case, a chromosome, a coordinate, a reference allele, and an observed allele. And you can see that this could happen in often two different ways, either from the actual genome itself, in the context of a chromosome, or it comes in from a transcript or a particular gene name. And I'm going to show you how you can even leverage just the gene name itself in Allele Registry for a biocurator use case. So the impact that this has essentially is that we're able to decipher the similarity, or basically the same observation in different contexts, whether it be the transcript, HCVS, we see here on the right-hand side, or the genomic context, or these other HCVS expressions that may be found in ClinVar, dbSNP, myVariant, Nomad, and others. And essentially what this is attempting to do is saying that all of these observations amount to the same identifier based on the mappings between the transcripts and the genomic builds. So here's some of the entities that we bring in to initially seed the Allele Registry: the myVariant.info, Cosmic, ClinVar, Exact, Nomad, dbSNP, and there's others as well. So here's what the UI looks like, and I'm going to show this in a little bit. It's basically at the very top, you start off with your canonical allele, the gene, a link out to the gene from HENC and NCBI. We have our link outs to other identifiers that were provided when we imported these to the API. We're actually able to obtain their identifiers and actually be able to have a link outs here, so that people will find that useful. We also have the JSON, the LD structure data here. And in the middle of the page, we have the genomic alleles, but the genome assemblies, and followed by the transcript alleles for all the transcripts that map to the corresponding genome assemblies. So the growth over the last 5 years or so is we're up over 1.1 billion canonical alleles, and our average API registration rate is about 50,000 registrations per second. And to put that in context, people are familiar with Nomad or the scale of Nomad. So at least 2.0, we were able to register the whole thing in about 7 hours. And one more point about this is it looks like it's flattened out, like people aren't registering, but in this period here, it was over 122,000 new variants are registered. You get these large leaps when you actually get these large data sets there that are upwards in millions, 100 millions. So here are some of our users. We get some decent use across the world. About 20 unique users register canonical alleles a month, and that varies from month to month, of course. About 250,000 visits to landing page a month, about 14,000 UI uses a month from about 100 countries, and over 1 million API uses per month in 25 countries. So the Allele Registry use. So we've had a handful of databases that have adopted the CAID naming convention, namely ClinVar, Civic, myVariant.info, Ensemble, database of pathogenic variants. We also have tools that have also leveraged the CAID, such as Link Data Hub, Variant Curation Interface, Evidence Repository, Functional Data Repository. We have a service level agreement with National Cancer Institute. And we've also supported numerous data aggregation efforts. All of us clinical laboratories are identify variants classified by more than 2 laboratories, so they actually can have some the different versions of transcripts and so forth that allow them to actually have one naming convention to identify the same variants. The VIC, the Variant Interpretation and Cancer Consortium, integration across 6 prominent somatic cancer variant knowledge base to register variants and aggregate these variant interpretations. The Australian Genomics platform, Shariant, also uses this, as well as Civic also uses this with coordinate lookup matchup with Allele Registry to see if they've observed something before. So here's reference databases I refer to, and the main points here is that we have over 700,000 transcripts, a good chunk of them from RefSeq and Ensemble. We update this data monthly, and we've also been working on appending these main select annotation labels within the UI, and I'll show that in a second. So the reason this is important, so when we actually talk about variant pathogenicity interpretation, there's some challenges that come with the variant curation, namely variant identity. And so the same variant can be defined differently in different sources. So the naming is just in that manner. There's also a diversity of evidence sources, lack of standard ways to actually accumulate the evidence, tracking the provenance of evidence used in interpretation, the transparency and auditability, which are key FDA requirements, and alerting users, curators to knowledge and content changes, which is an ongoing effort. Okay, so we see this as a four-step process, and Allele Registry is at the very bottom. So this is how we're building up to these, I guess, higher level functionalities. Starting with Allele Registration at the bottom, we can link to external sources for describing the same variant using that CAID. We can also excerpt additional data elements on top of that. And finally, we can disseminate the evidence via the evidence repository from curations that were published in the variant curation interface. So here's what the basic flow of the Allele Registry API, which does the vast majority of the heavy lifting in terms of registration, either retrieving and importing data from these data sources we mentioned before. We use APIs to bulk import this data. We've also seen users do single and bulk registrations, as well as single and bulk queries, and all this is done in APIs. For the interoperability parts, we have various tools, like Data Hub, Evidence Repository, Variant Curation Interface, and others that actually use the Allele Registry APIs to obtain this data. And we also have queries required for external tools and data integration outside of ClinGen. And so I covered a lot of this in Breakout session 3. So we also, everything is built in the APIs. I'm not going to show examples of these, but this is basically the idea is that everything you can see in the UI is also obtainable via API. Okay. So to get into the more novel parts of this presentation, as compared to the breakout session earlier this morning, is I wanted to do a use case from the biocurator's point of view. And so you see, we have this the publication in Human Mutation. It was a single molecule detection of cancer mutations using this novel assay. And within that, early in the paper, they actually described these different observations per these genes and their coordinates. One of the problems is that the HGVS expressions were not published. And what do I mean by that? So the HGVS expression identifies a specific chromosome contig, or, with the very specific name, or it actually has a transcript name associated with these genes. And so oftentimes we have people that want to register variants, but are looking at publications that are older or just don't include the specifics of this. And so what we've done is attempt to design tools to allow people the options to further explore what these variants might be, where the coordinates might be, and they actually have them registered so they can actually use their identifier. So, excuse me. So for this demo, I want to do an overview of the UI. I'd like to talk about new user registration, the different query options we have for small variants, the biocurator use case, which I just mentioned. Also, just let you know where the JSON LD structure is. And then after I talk about structural variant support, I'd like to show a demo if there's time. Okay. So here's the Allele Registry, and this is the splash page here. And first, I guess before I get too further, if I'm logged in, no, I'm not logged in. Okay, good. So if you're not logged in, if you scroll down, and again, this is reg.clinicalgenome.org. If you scroll down, if you're not logged in, it'll allow you to create a login. So I encourage people to do that. Oh, somewhere you log in, sorry. Once I log out, it'll allow me to do that. Okay. So if I go to create login, basically just put a username, email, affiliations optional, and the password. And then basically, if you click Allele Registry, it'll basically give you access to register variants, Allele Registry. So I'm going to go ahead and log in. Okay. Right to the main page here. Okay. And so a quick basic view of the UI. And so we have a lot of different query options here. Some of these are self-explanatory. Some of them are definitely aided by a supported search pop-up here. So if you click on this, it'll actually give you a little bit more information. So those of you that might not be familiar with things like HGVS expression, like what is that? What does it mean? We actually have some link outs to that. And it basically gives you a little bit more information on what this is referring to. And so the main gist here is that you have a reference specified here, basically saying this is a coding. You have your coordinate, and basically say your reference allele C has been, which is in your reference. Actually, you have observed T here. We also talk about these other entities here. I'm going to show you just a couple of examples of these live, but I encourage people to go through and read these. So one of the first examples we talk about is this canonical allele identifier. So we can search based on the CAID. And if it exists, it'll actually bring up this page. And again, like I referred to earlier, so we basically have the name up here. We have the link outs to the gene. for this gene here. We also have this link data here, and followed by the genomic alleles and the transcript alleles. And so we also have this main label for both the ensemble and the NCBI version. And so this main is this match annotations between NCBI and ensemble. So it's basically them saying that this is their preferred transcript. This may be oftentimes the newest one or the one that's actually agreed upon from this effort. And it's matched between NCBI and ensemble. So it allows people to kind of say, well, out of all of these, which one should I choose if they're all the same to me? And basically gives people a leg up in terms of what to actually select. Okay. As this HGBS descriptor, which is one of our most popular features, is people that actually have an observation in a particular gene or a particular chromosome, and they would like to know if something's already registered. And they can search for that. In this case, it is. It's the same example that we have for the CEID. But what we're going to do in a little bit is actually look for an example that has not been registered yet. Some of the other entities you could look for is ClinVar variation ID, dbSimp ID, exact, nomad, variant, or other identifiers. Also look for gene symbols, reference sequence and position, and this one's for copy numbers, and then I'll talk about it in a second. And then we also have some link outs to PubMed IDs. Okay, so in terms of news registration, query options, I'd like to talk about this biocurator use case. And so this individual read this manuscript and would like to actually curate these variants that they found in there. They found this paper interesting, and there's some interesting observations, some interesting data, and they would actually like this variant named. And so starting with this data, all we have is the gene, the coordinates, and those locations. So what we can use is this gene and variation query here, also known as these fuzzy queries. So I can type in the gene here, TP53, coding. It's not protein. I'll show an example of that next. Position is 743. A reference is a G, and the alternate is an A. So if I do a query for this, should return it. Okay. So what we see is that yes, this variant appears to already have been registered. We see that there's these different transcripts that have the same coordinates and that correspond to the same variant. So again, these things often require additional searching to make sure that it's not a coincidence. It actually is the same transcript you're looking at, and often this gives curators or other individuals an opportunity to follow up with the original publication authors to figure out if it's the same thing or not. But this is kind of like a step in the right direction for registration. And again, you can see all this link out data here. Using the same query, I'd like to show the example for this BRAF gene. Okay, so in this case, we're going to look at the protein version of this. We're going to look at this location, 600. Okay, so we're going to do a query based on the protein. And similarly, yes. So they found this location happens to be the same location, 1799 here. So this may or may not be the correct instance, but you see that we do have this identifier here that has already been registered. I'd like to show an example of an unregistered variant. So I'm going to look at the third example that was provided here. So you see here, this is a 34 position G to T. But what I'm going to do is I'm just going to make a slightly different version of that. I'm going to probably even do a larger deletion. Just so I can show you two things. One, I'm going to show you that you can use indels using the same tool, but also want to see something that hasn't been registered yet. So using that same functionality, this gene and variation based query, I'm going to switch over to indels. I'm going to provide this gene symbol. We'll do coding, and I'll say 35, just as something I know that probably doesn't exist. We'll say 50. I'll say a deletion in this area. So if we do a query, it says it's a valid allele, but it's not yet present in the allele registry. So what we can do is I'm going to show you in another window. We're actually going to query for this HGBS expression, which was just created by this tool. To show that it's a different functionality to get the same thing. So there's two ways to register this this allele. One is to click get identifier. And the second is to do it actually from that query interface that I was just showing you. So this can be registered here. But for this point, I'm going to show you this. If you were to do a query on the HGVS expression and it brings up a valid HGVS expression, which shows you all the overlaps between the genome builds and the transcripts, and you get to this page, and if you're logged in, you can click on Get Identifier. And so now this is registered. And so this count is pretty close to how many variants we have in the Allele Registry. So it looks like 1.139 billion. And again, you can use JSON-LD or these other link outs here. Okay, a couple things. I'd like to briefly mention CNVs, or what we're supporting for structural variants. And then I'd like to have some questions. And then if there aren't questions, I can actually show you some of that interface. Okay, so we just talked about is Get Identifier if you do a query and something does not exist. And so briefly for structural variants, so tools and resources for structural variants would lag behind capabilities that we've seen that is just for smaller variants. I have some stats here. I'm not going to get to everything, but on average, 2.9 rare occurrences at altered coding regions. And we've seen as we go forward that the lack of support for these entities and account for about 17.2% of rare alleles worldwide, but 90% are non-coding, 3 times more likely to be associated with a GWAS study signal than SNVs. And these large ones are more likely to affect the expression of gene compared to an SNV. So these do propose some challenges, especially like in terms of computational, let alone the actual the mechanisms actually that do detect these. So detections improve, which result in multiple useful databases. And so what we've been exploring so far is ClinVar, DBVar, Nomad, and NextSec. But there's not really, nothing exists right now that's a dereferenceable naming convention in which to identify these single structural variants across genome builds, transcript versions, in the same way that we attempt to do for the small changes that we've seen representing by CAIDs. So we also see, so stage 1 is actually the copy number support, which we've supported. And what we're working on right now is ongoing is non-CNV support. So our goal is to extend the Allele Registry continually to support these, and so that the other curation efforts can actually leverage the naming convention in the same way that they use CAIDs. A brief mention on this. So just something that helps me for folks that aren't very familiar with this is just what's a structural variant, what's a copy number variant. So structural variants are variation in the structure of an organism's chromosome. They're between 50 and millions of base pairs long. And this is debated and changes all the time, but they're generally larger than the small ones. But it really depends on what database has them. That's kind of how we're viewing these. And so copy number variants are structural variants of types of deletions and duplications in the genome. And then so CNVs versus structural variants. So copy number variants are unbalanced structural variants with a gain or loss of genomic material. So that's something that kind of helps us describe these and allows us to create this descriptor that we're able to use for registration and querying. And so what this is what the descriptor looks like. It's kind of a little synonymous, but a little bit with HGVS expression, and similar to what was enumerated by ClinVar for describing these. So it's kind of a hybrid model between these different published models. Also the ISCN and ClinVar preferred name. And so this is the total number we have right now, about 344,000. So this list is growing, and we have this endpoint here that actually tracks that. I'm not going to show that now. Okay. And so what we've done is that we've also spec'd out non-CNVs structural variants, and not to get into these details here, but these of type deletion, duplication, insertion with source, and all the way down to inversions, and then these complex scenarios. Okay, I want to leave 6 minutes for any questions that folks might have. Anything you'd like to see in the Allele Registry? Any examples? And if there are no questions, I could show a couple more things. And I'm going to leave this acknowledgements up. Thanks everyone for all their hard work.
Can you go back to the fuzzy search example?
Which example?
Fuzzy search, gene to HGVS.
Mm-hmm.
Right, and let's say select BRAF, and let's say the protein, and let's say 600. Now, if you click on select one, so what would be very nice and useful for people if addition to 3-character code, just in parentheses, you would add just 1-character code. Because often people, of course, use it in the hurry, have limited amount of time, and having just single letter notation for amino acid edit would be very useful for many people.
That's a very good point. Yeah, I think that's probably the more common use case. Absolutely. I don't think that should be a problem. Yeah, no, thank you for that question.
Thank you.
Yeah, this is Jenny. That sounds like a great idea. We do have another question in the chat as well. Please discuss present and future support for haplotype identifiers.
Yeah, so we have some support for haplotypes existing in Allele Registry. I'd be hard pressed to find a good one in a short amount of time. Sorry about that, for haplotype identifiers. Yeah, so there's some ongoing work that we actually need to continue to do to support what we are considering a genome build and what it means to have something different from your standard diplotype versus haplotype. Yeah, if you don't mind, if you write your if you could put your email address down there, I'll be able to send you something after that. I'll look it up during the next talks. But yeah, I can give you a couple examples though.
And another question, and you may have mentioned this, but do you support large indels with unknown breakpoints? Yes.
And so what I didn't get a chance to really talk about in this descriptor, if I understand your question correct, we do provide these precise or questionable ranges between the start and stop points. And that is for CNVs. It really depends on if it's a type deletion, we're still working on that. But if it is copy number based, we do provide that. But if it's non-CNV, that's work in progress. But that's definitely on our radar because that's a lot of these observations. They don't know this, and then the result of a lot of these experiments are imprecise.
And I think this is a comment. Lack of bulk downloads remains a critical weakness and prevents numerous use cases. So could you comment on that, please, Kevin?
Sure. And so the way the Allele Registry works is, I kind of mentioned this in the breakout session earlier, is that it's kind of a living, breathing thing. Is that I made the point is that there could have been 200 variants that were just registered, since I started this talk, and so it's vastly growing, and again, even a certain amount of time. So it's possible, and I'll give it some thought, and I'll talk with our dev team to see if we want to maybe do some freezes, monthly freezes or something like that, that allow people to get data in via FTP type of route or something. But everything we've done in the past is exposed via APIs, and people can actually do these queries. And generally, we've had success with people getting what they wanted, whether it be all the variants for a particular gene, for a chromosome, for a chromosomal region, or really everything, just looping through one by one. That's kind of the most efficient way to get things, because things are constantly changing. Identifiers are changing, not only adding things, but we're actually, these things are getting tweaked, and things that, certain link outs have changed, between ClinVar, they retire certain IDs. And so it's kind of this, mutable thing that is kind of hard to nail down. But that's a good point, and I'll see if there's something we can do to do it like some sort of dump. But yeah, I appreciate the questions.
Great, thanks so much, Kevin. So we're at 4:10 now, so I think it's time to switch over to Christine.
So can you hear me and see my slides?
Yep.
Thanks, Jenny.
Yeah.
All right, and thanks, Kevin, for that really lovely talk. So hello, everyone. My name is Christine Preston, and I'm delighted to be here to talk to you today about ClinGen's Variant Curation Interface, or the VCI as we call it. So the VCI platform is used by biocurators as part of the ClinGen variant curation process. And curation work is done in the VCI by approved ClinGen variant curation expert panels, or VCEPs, and this is then disseminated publicly. And I'll talk more about the dissemination part later on in this presentation. So first, we'll do some brass tacks stuff. So the production version of the VCI can be found at curation.clinicalgenome.org. And this site supports both the VCI and also the Gene Curation Interface, or the GCI. For most of this talk, I'm going to focus on the VCI, and Matt Wright will be presenting on the GCI directly after this. Now, the first thing to mention is that there's actually two web addresses where both the VCI and GCI functionality exist. Now, the production site URL is the same. It's curation@clinicalgenome.org. And then we have a demo or test site, which is at curation-test-clinicalgenome.org. Now, the production site is intended to be used only for real curation. All formal curation must be done on the production site. We have the demo and test site because it's intended to allow users a space in which to explore and use the interfaces. It's very important to note that on the test site, curation data is periodically wiped for the site. This is in part to encourage people to not use it as a curation for actual curation work. And we recommend that all production users register for both sites. It gives folks a place to practice when they're learning how to use the interfaces. How do you register for the sites? For both sites, you register by filling out a registration form, and you'll have to do them independently for both the production and the demo or test site. To register, you'd click on this login button at the top right of the screen. This will then take you to a sign-in modal, which you will use to sign in once you have an active account. Now, to register for a new account, you'll click on the Create Account button, and this will take you to the Create Account modal. If you fill out the modal, you'll eventually receive a email verification code. And once you use that verification code to verify that you are indeed the email you've registered as, that will finalize your registration. I do want to note that on the production side, we add users by administration approval, and this happens every few days during business days. So initially, depending on when you've registered, you may receive a thank you for registering screen. You can always email us at clingen-helpdesk@list.stanford.edu to fast track an approval if you really need to get in immediately. Once you're approved, you'll then be able to sign into the site using the sign-in modal. And the demo and test site is a little different. It's completely self-serve. So once you've registered, you'll be able to access the VCI within the demo site immediately. Okay, so now when you first enter the VCI, you'll land on what we call the VCI Dashboard page. At the top, as shown by the red arrow marked in 1, is the welcome banner. This lists both your name and also in parentheses, it'll list if you're curating as part of an affiliation or as an individual. And I'll talk a little bit more about affiliations in a moment. Now, the red arrow marked in 2 is kind of a navigational tool. You can access a summary list of all variant interpretations that are available within the VCI. You can return back to the dashboard, and you can also see a summary list of all gene disease records in the GCI. The number 3 arrow shows the variant table. This displays all the variants that has an interpretation record for this affiliation, or if you're curating as an individual, for you as an individual. As you can see, it lists the variant title, any associated disease terms and modes of inheritance that have been curated along with that variant. It includes the status of the record, the classification, any criteria that have been evaluated as met, and any modified strengths where applicable. And it also shows the date created and the date last modified for all the variants listed. This table is searchable in a couple of different ways and also downloadable in a CSV format. And really quickly, I want to talk a bit about how affiliations work in the interfaces. So affiliations are a group of users who curate genes or variants together within the interfaces. Now, all members of an affiliation have the ability to initiate, edit, and change the status of all curations owned by that affiliation. This includes approving and publishing curations. GCI users must be part of a gene curation affiliation. But the VCI supports both ClinGen affiliations and also community group variant curation affiliations, and of course, individual use as well. If you're part of an affiliation, you can select to curate under that affiliation or as an individual at any time. And to change affiliations, just click on this Change Affiliation button shown here on the far right. You'll be shown a drop-down which lists all affiliations you are a part of, and you can select from this, and then that will appear once you return back to the dashboard and the main interfaces. So now I'm going to talk quite a bit about the VCI workflow and the support within the VCI for the ClinGen FDA recognized curation workflow. So the VCI is an integral part of the ClinGen FDA recognized curation workflow, and it's designed to support the ClinGen workflow from identifying the variant all the way through to disseminating the final approved variant classification. And the first step in the VCI is, of course, to identify the variant. To do this, you can click on the new variant curation button highlighted here by the red arrow. This will take you to the search and select variant modal, and you can search for a variant using either ClinVar ID or an Allele Registry ID. And Kevin just gave a really nice talk about the Allele Registry, so hopefully that answers some questions about that. Once you have selected your variant and go in and move along, you will then be shown into the evidence view. So the evidence view for the variant you selected is a status where you haven't actually started interpretation record for the variant. You're able to simply see all the evidence available in the VCI for the variant. And as you can see here at the top, the VCI links out to a number of different locations. So the VCI is intended to be a rich curation interface, and we bring in a ton of data, evidence data, and also links out to various different resources to try to create a really rich and useful curation environment. The VCI at the very top, you'll see, will list various variant ID sources, such as ClinVar variation IDs, ClinGen Allele Registry IDs, dbSNPs, and links to external resources, such as the UCSC and Ensemble genome browsers. Additionally, as you can see down below the evidence view, there's 6 different tabs within the VCI, and the VCI is set up within these 6 tabs from basic information, population, variant type, which is shown here a bit, experimental, case segregation, and then gene-centric. All of these different tabs have a rich set of evidence from a variety of sources brought in and displayed for the variant. Many folks use this evidence view functionality just to take a look at different evidence that the VCI pulls in for a given variant when they don't want to actually do curation work. Now, if you wish to start an interpretation, then you would click on the interpretation button here, shown here by the red arrow. This will first ask you to agree to the ClinGen submission policy agreement. And in particular, I'd note that this spells out that the data in the VCI may be made publicly available and that no publicly protected health information or PHI should ever be entered into the VCI. At this point, you've started a variant interpretation record. And in this experience, users can see both all the evidence and resources that the VCI brings in, but also they can manually add evidence and do curation work. You can do things like add a disease and a mode of inheritance to the interpretation. And there's also, as you can see, this bar right here is the criteria bar, which indicates which criteria have been evaluated as you move along the curation work. And then shown here is a subset of the basic information tab. All of these images are going to be partial. The VCI is long and skinny, and slides are short and fat, so it's a little tricky to get everything in. But basically, you can see that there's a ton of information just in this basic information tab, such as ClinVar interpretations, variant names and molecular consequences, which aren't shown here, and other interpretations for the variant within the VCI. Moving along, you can see that, for example, in the population tab, we bring in a number of different population resources, including Nomad. And I just mentioned that the 4 middle tabs, population, variant type, experimental, and case segregation, in all of these tabs, curators can both view evidence and also will do the evaluation of the criteria codes within these groups. So this is the tab where you can see not just Nomad, but other population data sets as well. And this is also the tab where curators will evaluate the 3 population ACMG criteria, BA1, BS1, and PM2. So again, the VCI brings in a rich set of evidence from a variety of sources, and we're constantly working to add additional evidence sources. Here you can see some of the data brought in for the variant type tab, including many in silico predictors, conservation analysis scores, and links to 4 different splice site predictors. Now, I'd also like to mention that, of course, some of the evidence is never going to be programmatically available. So I don't have the workflow here, but the VCI allows curators to add their own evidence, such as PubMed IDs. And then that's displayed and added to the evidence repository so that other curators looking at the same variant will also see the evidence added. So once again, this is a community curation tool. I'd like to mention that 2 of the tabs have specialized options for adding evidence. Structured functional evidence can be added to the experimental tabs by the functional data pipeline, which isn't shown here. And the case segregation tab has a special set of forms that can be filled out, resulting in a structured view, which I tried to show here, where you can view all the evidence by criteria across different evidence sources. And again, since the evidence evaluation is a key part of this process, each ACMG criteria is shown in its relevant tab alongside the relevant evidence, and curators can then make their evaluation. So as you can see here, these are the population criteria codes that can be evaluated. As curators work along the criteria evaluations, the criteria bar will visually display which criteria have been evaluated. Colored criteria boxes indicate that the criteria has been evaluated as met. White indicates not yet evaluated, and gray indicates the criteria is not met. Finally, users can keep the auto-calculated classification, or they can override it and manually input a different classification if determined necessary. Curators can write in an evidence summary as well. Finally, I wanted to talk briefly about the status of variant interpretation records in the VCI, as this also supports a general workflow. We have 4 statuses shown here in these colored buttons. And these are displayed in the same manner as in the GCI. The in progress status indicates that curation work is ongoing and not yet complete. Well, the provisional status is used by curators to indicate that the curation work is completed, but that this still requires expert review, which is part of the workflow. The VCI maintains a data snapshot for any saved provisional or approved interpretations, and this can always be accessed by curators if desired. Finally, there's approved and published statuses. Approved is a status indicated that a record has undergone expert review and has been approved by the VCEP. Published is a co-status to approved, where the VCI has been published to the evidence repository. And the VCI supports ClinGen VCEPs in publishing their approved interpretations to both ClinGen's evidence repository, or the eRepo, and also we provide ClinVar submission data. And as I said, the VCI curations can be disseminated to the eRepo for public dissemination. And I believe there's going to be a nice talk on the eRepo tomorrow at Breakout session 7. So please see that if you'd like some more information. And then the VCI also supports users in sending their interpretations onto ClinVar. This is still a somewhat manual process, and we're actively working on making it a more automated process in the future. And I'd also note that approved ClinGen VCEP interpretations from the VCI are noted in ClinVar as coming from an FDA recognized database. And I think there's a ClinVar submission session, but I think it's concurrent with this one. And then here's another image of some ClinVar data from the VCI and the P10 variant VCEP. So we're very proud of the current VCI, but we also recognize the need for scalable curation workflows. And I just want to say that the VCI has seen substantial growth in both curators and curation since it first launched. As of this week, we have over 8,300 variant curations in the interfaces and over 1,600 users with an active login in the production size. But we really are looking towards the future in terms of scalability. And so very briefly, within the last few minutes I have, I wanted to discuss an exciting new feature we have launching in July of 2021 called the Variant Prioritization, or VP. This feature is a building block in our long-term plans to create scalable workflows to support bulk curation work within the VCI. Now, the intent of the VP is to provide large-scale variant assessment, eventually supporting bulk curation workflows. We currently have a beta version supporting 5 genes, and the Stanford Baylor development teams are actively testing another 106 genes to be added to the VP. We are currently prioritizing genes that are being actively curated by ClinGen VCEP. And I'd like to talk, the VP has a number of featured planned, but I'd like to spend the rest of my time here talking about one of the main features that users will be able to use at launch, which is the filtering workflow. So in the filtering workflow, users will be able to search for all variants known to the linked data hub by a gene name. And they'll then be able to filter down the results by a number of different filters. So for example, they can filter based on select ClinVar data, VCI status, molecular consequences using the main transcripts, nomad population data, and Revell scores. This data is supplied by the linked data hub, or the LDH, and please go see Kevin's poster on the LDH tomorrow. I believe it's in poster room 4. Some of these filters are quite sophisticated. For example, you can see a population filter here for some nomad data. This supports filtering based on population data, any population data, and also data based on allele count, number, frequency, and the number of homozygotes. Additionally, biocurators will be able to save these filter settings. and they'll be able to reapply the filter settings if desired. So if a very complex query is set up, it can be saved and used later. I'd finally like to mention that biocurators can also define what information is displayed associated with the filter results. And the VP, of course, is intended to support bulk curation workflows in the VCI in the future. Our development plans for it include supporting transfer of VP identified variants to the VCI for full curation, as well as bulk curation workflows. So with that, I'd like to take a minute. There's so many people who have contributed to the VCI. It'd be very, very challenging to add all of them to an acknowledgements thing. But I do want to talk about a few folks. Our Stanford team here, we have wonderful leadership with Carlos and Terry. Matt and myself are the product management sector of the team. And then we have a really fantastic software engineering team led by Rao that do all the hard work putting together these beautiful features. And then I'd also like to take a moment to acknowledge our 5 variant curation stakeholder proxies, Marina, Jenny, Natasha, G, and Justine. They give up time each week to talk about new features and give their input in terms of the developmental process. And we're very thankful to them for that. And then finally, I'd like to mention how anyone can get help, because we have a number of resources. Actually, within the interfaces, just on the right-hand side, there's this header that lists a whole bunch of different help documents, including our wiki that we maintain, Matt and myself, for the GCI and the VCI, as well as the SOPs for the GCI and the VCI from the ClinGen group. We also have our terms of use listed there. And you can also email us. You can email for the VCI. There's VCI@clinicalgenome.org. For variant curation, there's variant curation@clinicalgenome.org. And you can always email ClinGen as well. So that's all I have, and I'm happy to take questions.
Any questions for Christine? Christine, it's Jenny. I wonder, do you want to talk a little bit about some of the future plans for the VCI, like including VCEP specific information in the VCI?
Yeah, I think that's a great, so I don't know how many of you were able to make I think the C-SPEC talk is tomorrow, perhaps. Anyways, in conjunction with Baylor, we're putting together something called the C-SPEC. And I know Stephen and folks talked about it a bit this morning. And it's going to be a really, I think, great opportunity for us to take some of the specifications being made by various VCEPs. The initial plan is to allow people to associate a specification with their interpretation and also display that in the VCI as people are doing the curation work. But eventually we'd like to use it more to support potential automation. So really helping people to find automated ways to evaluate various different criteria. So I'm excited about both that and the VP. I think are really great opportunities for us to expand the speed of curation and create more automated workflows where it's applicable and appropriate. Thanks for that question, Jenny.
Any other questions for Christine? So do you think we should move on to Matt?
I don't know. It depends whether you think people might move across to this session.
Right. Yeah, you're right, Matt. Maybe we should hang on until 4:40. So we have about 10 minutes. So maybe do folks have anything particular they want to bring up about the interfaces? Any questions that they've had? Any improvements that they think could be made as you've been doing your curation work?
Joanna has actually just put a question in.
Oh, yes.
Yeah.
Thanks, Matt. So yeah, she says, this might be a small issue, but in the in silico tabs, it usually appears several interpretations, which I assume are related to different transcripts. Is there a way to define the correct transcript to use in evaluation?
That's a really great question. So right now, within the VCI, we're very genomic focused for the variant. And that allows us to identify the variant in most locations, which is very important, but it does create challenges, for example, with like molecular consequences and things that are related to transcripts. So we, as part of the CSpec, we've discussed, although it's definitely not going to be a V1 feature, adding the option of defining a transcript. It would be probably part of the CSpec registry where you could define a specific transcript. I do think that would open up some opportunities in terms of automation, because then we'd know a little bit more about the appropriate molecular consequence. There's also the opportunity, I think, to potentially leverage the main project to get a better sense for transcripts. And in fact, within the VP feature, because we do have a molecular consequences filter, that filter is going to be exclusively on main and main select transcripts. So we definitely are aware of the need to have transcript level support, but we're still kind of figuring out some of the details. I hope that answers that question.
Oh, Joanna said, thank you. Well, thanks for the question. This is really helpful. I wonder, do you think it would be helpful, Christine, to talk about some of the developments that are planned to be made? Things like tagging, ClinVar submission, that type of thing.
Yeah, we can. That's a great idea, Jenny. So we definitely could talk. Let's see. So what should we talk about? So I think the one maybe we should start with, which I'm very excited about, is we're actually planning on launching very soon the audit trail, which I'm very excited about, because we currently do actually maintain in the back end of the software, essentially an audit trail. So if we ever needed to figure out like who had done what evaluation at what time, we could go back into our data system and identify all of that. That's been true from the beginning. However, we're about to expose that at the level of the curation records to our users. And so when you go in, you'll be able to see, and this will be retrospective, it'll be for all curations that are currently in the interfaces. You'll be able to see when saves were made, essentially. So when the curation was started and by who, when criteria were evaluated and by who, and how they were evaluated. So if changes were made, you'll be able to identify that. So there will be, as I said, it's an audit trail that can be just listed within each curation record. So because it's a community curation tool, and because we have, affiliations, many that go on for years, we're hoping this will help folks, if they have questions about what happened, go and look at that. So that's something that we're planning on launching pretty soon. Most of the development work on that is done. As I said, the VP is something else that we're launching in just a few weeks, really, and we're very excited about that. In terms of the future, definitely a lot of our plans involve support for bulk workflows, such as the CSpec. We also have things like we want to do evidence, what we call evidence specification. So there would be more of a programmatic link for curators to set up between the criteria evaluated and the evidence used. So right now, I think people are writing it down in the explanation, which is obviously very important, but this would allow for that to be programmatically linked. For example, if you were evaluating a population criteria, you could indicate what data set within Nomad or whatever your favorite population data set is that you were using. So that's a building block also to some of our future automation plans, because once that's there's that programmatic link, then not only will I think that be very exciting in terms of the dissemination, that's something that we could pass on to the e-repo in a programmatic fashion, which could allow for some great data mining opportunities. But also it's something that could be used upstream of the VCI, for example, in the VP, where folks could identify using those filters in the VP, what their metrics are. For example, I think the population ones are the easiest to talk about, although I know from a bit of personal experience that they're still very challenging. But you could imagine that a VCEP could specify, you know, this is our cutoff for BA1 or PM2 or what have you, do the filtering within the VP on a gene that they're interested in, and then have that information pass on to the VCI in a structured way such that those could be auto-evaluated. And you can imagine there might be some where maybe you wouldn't auto-evaluate a criteria, but you would auto-add the evidence or programmatically add the evidence within the interfaces once it had been identified within the VP. So again, a lot of our focus is, from my perspective, is in terms of both making a more personalized experience so people can get more guidance from the VCEPs and all the great work the VCEPs have done in terms of coming up with specifications, and also doing things in a more looking towards a future where there's more automation to the system, where there's less clicking and, you know, finding the source and adding the source kind of information needed. Also, one thing which, you know, I'd love to do more on, but we haven't quite figured out the right approach, I think, is support for identifying publication resources. And I think that that's something that it would be great to have. So there's, I think, a lot of really awesome opportunities. Oh, and another feature I'd like to mention, since you asked, Jenny, is we're working closely with the Baylor team for the Linked Data Hub to be able to bring in a bunch of external resources. And so the idea is that there's a number of different databases out there that have you know, really rich data sets. And we'd like to be able to, at least in some way, indicate within the, I think the V1 would be, we would indicate in the VCI that there is this information and tell people where it is regarding the variant to these external databases. But in the end, we'd like to be able to bring that information, those pieces of evidence in those other databases into the VCI. So it's a collaborative project between our team and Baylor LDH team. And then also we're talking right now with a number of different groups that are, you know, building and maintaining these various different databases to try to figure out the best way to do this so that it's a structured workflow, and so that it kind of, we don't have to reinvent the wheel every time there's a new database that we want to add additional information to. So I could talk about development for a long time. You should cut me off.
There's lots of exciting things.
Lots of exciting things happening. Yes.
So it's really great, and of course, the VCI team will keep everyone up to date with what's available in new releases, and also on the BioCurator working group, making sure that everyone is aware of anything new. So, but I think I speak for lots of folks when I just say I really appreciate all the work that goes into the interfaces and all of the informatics for ClinGen. So thank you, everybody. So thanks, Christine. So we'll move on to Matt now.
Yeah, so I've got 1 minute, but so in that, I'll use that 1 minute to say, well done, Christine, for filling that time with your vision of the future of the VCI. Very impressive, thank you. So hello, everyone. It's with great pleasure that I follow on from Christine's excellent presentation that you just saw on the VCI with the pivot to the other curation tool developed here at Stanford University, the Gene Curation Interface, or GCI for short. And my objective today is to walk you through the gene curation workflow that takes place within the GCI. I have to tell you, when this is normally presented, it's in like over the course of an hour normally in a training thing, so this is a very abridged version. As you no doubt all know by now because you're attending this meeting, but ClinGen is an NIH-funded initiative dedicated to building a publicly available genomic knowledge base of curated information in an effort to improve patient care through genomic medicine. Now, a critical component in building this genomics knowledge base is evaluating the clinical validity of evidence implicating genes in human disease. In order to assess the clinical validity of a gene-disease pair, one must consider how strong the evidence is that that variation in that gene causes the disease in question. As one of its goals, ClinGen's Gene Curation Working Group has developed a framework to standardize the approach to determining the clinical validity of a gene-disease pair. So this framework defines the criteria needed to assess clinical validity. It quantifies the evidence supporting the gene-disease association, and it allows the curators to methodically classify the validity of a gene-disease pair. And this original framework was described in the 2017 Strand et al. Publication in the American Journal of Human Genetics. Further information on the ClinGen gene curation process can be found in the gene disease validity section of the ClinGen website. Here you can find educational and training materials, and you can view the results of gene curation. Additionally, the current specification of the framework that the GCI supports can be found in the standard operating procedure section in the SOP. And if you need help in gene curation, you can reach out to the Gene Curation Working Group via their email genecuration@clinicalgenome.org. Here on the left is the workflow for the ClinGen gene curation process. And as you see at the top, it actually starts with pre-curation. That's gathering data from relevant papers with a literature search, and importantly, assigning the correct disease using the Mondo disease ontology by following ClinGen's lumping and splitting criteria. And Courtney Saxon has a session on that, which may actually be happening at the same time as this one. I can't remember which session it's in. But the Gene Tracker, which is Courtney's tool that they've developed at the UNC, is the tool that assists curators in this pre-curation process. And you can find out more information about this on the ClinGen website. The Gene Curation Interface, the GCI, is on the same platform, as Christine said, as the VCI at curation.clinicalgenome.org. And it was developed to support the ClinGen gene curation process, as described in the Gene Disease Clinical Validity Classification Framework. And we keep the GCI closely aligned with this framework. And so, for instance, if a gene curation SOP update requires a new feature to support it, then we update the GCI accordingly. Following the framework, as you can see on the left-hand side here, the GCI supports curators in curating genetic and experimental evidence. It allows them to assess and score that evidence, and then provide a classification for the validity of the gene disease pair. The classification is then expertly reviewed before being saved as final approved classification, which ultimately gets published to the ClinGen website. Now, unlike the VCI, which is a community curation tool, the GCI is only used by ClinGen gene curation expert panels, known as GCEPs. And members of these GCEPs can all curate together within the GCI as a single group known as an affiliation. And Christine described to you a moment ago how affiliations work within the interfaces. To get started in creating a new classification, a GCEP curator must first create a gene disease record by clicking on the new gene curation button in the header. Below that, you can see the affiliation you're logged in as within the dark blue banner, and also within parentheses after your name. The record will be created under that GCEP, so you need to make sure that you're in the right one. The three essential pieces of information to create a record are the gene name, the disease, and the mode of inheritance. GCEP curators will have gathered this information as part of their pre-curation process in the gene tracker. The gene name is specified by adding an HGNC gene symbol. We provide a link out to HGNC to check your symbol is correct, but the field also checks to make sure the gene symbol is valid when you go to save a new record. For the disease, the GCI only accepts Mondo disease IDs. We provide a helpful pop-up modal that allows you to search the OLS ontology lookup service, which is the EVI, with your Mondo ID to make sure you're entering the correct disease. And finally, the mode of inheritance can be selected from a pull-down menu of modes of inheritance. Now, this selection will affect how your evidence is scored within the GCI based upon the scoring stipulated for that specific mode of inheritance within the SOP. This was actually recently updated, so the GCI recently now allows scoring and publishing of mitochondrial genes, and the SOP will be updated soon to reflect that as well. If you wish, you can also add an optional adjective to the mode of inheritance with selections also provided by a pull down. For instance, here are the options for autosomal dominant. And then once you've actually started an entry, you'll see something like this. And you should check in the banner at the top that you've put all the details in correct before you continuing with your curation. I should say this and all the following data you see in this presentation is actually not real data. This is just screenshots that I made up on our test site. And next, once you've got to this step, your first thing you'll need to do is add papers from which you're going to source your evidence. At this time, you can only add published data via PMIDs, but we're looking into being able to add preprint articles via DOIs as well, such as from medRxives. And as you can see, again, we provide a helpful pop-up modal to help you search for the correct paper. Make sure you're adding the right one. When you add a paper, information about that paper appears in the center panel, as shown here. If you have the option, you have the option of providing further information if you want, general information about the paper, such as whether there was scorable evidence or not. I can say, an example of that, for instance, might be if you had a review article that helped you understand the gene-disease relationship, but it doesn't actually have any case or experimental evidence that can be assessed, but you could mark that in as a non-scorable paper. For each added paper, you can now curate in the evidence you found in that paper. As you can see on the right-hand side here, the evidence you can curate is split into genetic and experimental evidence types. Genetic evidence is further split into group, family, and individual case evidence, and also case control. Just to be clear on the distinction between those different genetic case levels, you should use group when there are many probands with similar characteristics found within a single paper. You should use family when you want to document specifics about multiple family members, or if you want to score segregation information. And finally, you should use individual when you want to report a single proband on a paper. Whichever evidence you choose to add, the GCI guides you in how to add the evidence you need according to the gene validity framework. For instance, here you see the top of the group evidence form and the fields that are options for adding in disease and phenotype and demographics information about that group. The forms allow you to capture evidence in a structured way according to the data requirements of the gene validity framework. And again, where possible, we provide pop-up modals to look up and bring in the correct information. For instance, when you add in human phenotype data via HPO terms, you can look up to make sure you're adding the right ones in. Now, you'll notice that so far we've added in the disease using Mondo ontology terms and now phenotypes using the HPO ontology terms. That's because wherever possible, we encourage the collection of data via ontologies. That's because using ontologies enables each term to be precisely defined, and it allows cross-analysis of comparable data from different sources. And as such, it then provides a much richer data output for analysis afterwards. Additionally, where we're able, we offer the option of copying evidence from one location to another within an entry. So for instance, at the top of this family evidence form, there's multiple options to copy across the evidence that was already saved into the group evidence form. Okay, voila. Now looking at this family evidence form, so far we've just been looking at the top of the form. This form is actually much longer, it's got other sections beneath it. Below this section is a section for collecting information on the methods identified in the paper. And then below that, a section on collecting information on the test individuals within that family. Now you'll notice that there's an asterisk next to that first field. Any field with an asterisk indicates that it's a required field according to the SOP. And so if you go to save the form, like I want to save this form now, it would provide me with an error message because any field that has an asterisk can't be left empty when you go to save the form. Yeah, and so they've seen out at the bottom of this form, and at the bottom of each evidence form, there's the option to score the evidence that you've curated in the form above. For family data, if you filled in all the test individual stats, that is the number of affected and unaffected individuals in the family, then the GCI will automatically calculate an estimated log score for you. However, you also have the option of just using a log score from your publication instead if you want to. Either way, you need to specify if the sequencing method was from candidate gene sequencing or from exome genome sequencing, because this information is used by an algorithm in the GCI that was devised by the Gene Curation Working Group to convert your saved log scores into points. And it's these points that will then be used toward calculating your final classification. But before we move on to classification, let's look at the curation of other evidence types. The individual genetic evidence form has many of the same fields, group and family. You're required to specify if the individual is a proband or not. I think I've gone 1, 2, 1. There we go, yeah. Or not. And if you want to go to score your proband, then you need to add at least one variant. Here we go, adding the variant. For this, GCI has another helpful pop-up modal. This time to help you make sure you're selecting the correct variant. And variants can be added using either the ClinVar variation ID or a ClinGen Allele Registry ID. Again, when you go to the bottom of this form, you have the option to score the evidence. You also have the option to select review if there is something about this evidence that you want to be reviewed by experts, or contradicts if this piece of evidence contradicts the gene disease association. However, if you select to score the evidence, then you'll first need to select the case type. And as is the case for all scoring in the GCI, based on your selections, you're provided a default score for that evidence. However, you can choose to override that default score if you wish, up to a maximum of points allowable for that evidence type, as long as you provide an explanation for your decisions. Now, as you can see on here, I've actually labeled this with a big red SOP v7, and there's a reason for that. It's because the SOP was recently updated, and it's actually now on version 8. And this will affect how individuals are scored in the GCI. Within SOP version 8, the current SOP, you no longer score each proband, but instead score each individual variant associated with the proband. The GCI will be switching over to accommodate this scoring change at the end of July. When that switch to SOP v8 happens, then at the bottom of the individual evidence form, each time you add a variant, there are required questions you need to answer in order to score that variant. As you can see at the top here, these include the variant type, is it de novo, and if so, whether it's maternity, paternity confirmed, and is it supported by functional data. And then based on your answers to these questions about the variant, you'll get a default score for that variant. And in cases like the autosomal recessive one here, the scores from both variants are then combined to give you one score. If you'd like a full explanation of the changes in SOP version 8, please refer to the documents available on the ClinGen website. The final genetic evidence category is case control. Within the GCI, the case data is entered on the left-hand side and the control cohort data on the right-hand side so that you can directly compare them side by side. At the bottom of the evidence form, you can provide an evaluation of the evidence, including any statistical analyses of the evidence. And again, at the very bottom, you can score the evidence. And then finally, this is the last piece of evidence. Finally, experimental evidence. This is split into the 6 different types you see here. And depending on your selection, you see different forms to enter that specific type of evidence as specified in the SOP. All the experimental evidence forms adapt to your selections within the forms. For instance, here is the top of the model systems evidence form. And depending on whether I select non-human model organism or cell culture model, the form fields change to reflect my selection. So in this way, you can precisely capture the required data for each subtype of experimental evidence. Again, we encourage the collection of experimental evidence via ontologies wherever possible, and you'll get warning signs if you enter these incorrectly or if you leave them blank. You can associate a variant with your experimental evidence. And then once again, once you've finished curating in all your evidence into the form, you can score your experimental evidence at the very bottom of the form, as shown here. Now, once GCEP curators have curated, assessed, and scored all the genetic and experimental evidence types, that are associated with a gene disease pair, the GCI takes all those scores and tabulates them into the calculated classification matrix. The genetic evidence is at the top half of the table and the experimental evidence at the bottom. I should say this is the SOPv8 matrix, and this will of course be updated when we switch to the SOP version 8 at the end of July to include variant rather than proband scores. The segregation evidence, as I said earlier, the stable odds scores are converted to points in the table. There are also some rules embedded within the functionality of this table that only allow certain evidence types to reach a threshold of point, so like a maximum number of points. The allowable points totals are summed together to produce the overall calculated total points for the gene disease pair. These points then advise the calculated classification. So for instance, this example has only 3.25 points, so it's calculated to fall within the limited classification. On points alone, calculated classification can go up to strong, so long as it has at least 12 points. However, in order to bump up to definitive, the evidence needs to be shown to have been replicated over time, in which case you can check the box indicated and bump the calculated classification up to definitive. You can always opt to change the classification, but again, you'll need to provide a reason why. The evidence summary text, the second arrow there, is also required to save a classification. We have a link out to information on the website on how to structure your summary for this classification. And it's important to get that right because upon publishing, it will be viewable publicly on the ClinGen website. Upon saving a classification, you can now submit and save that classification as provisionally complete. This step indicates that the GCEP curators are finished reviewing and assessing the evidence and that they're ready for the saved classification to go out for review. To help in this review process, an evidence summary table is provided within the GCI that shows all the evidence that has been assessed by the GCEP curators for this entry. Again, this isn't real evidence. A real evidence summary would actually be like extremely long. You can imagine when it has all the evidence in it, containing every bit of like assessed evidence, including all the pertinent information collected for each piece of evidence and how each piece of evidence was scored. We provide instructions on how to export this in PDF format so that it's easy to share, so that you can share it amongst all the members of the panel who are all the experts who are reviewing the classification. Once the review is complete and assuming all is good, the GCEP curators can go on to finally approve the classification to mark it as final and complete. You can select your own final approval date because we recognize that the time when a curator enters the final approval into the GCI might be sometime after the expert reviewers have met and finally approved the classification. During the approval process, you can choose to recognize other groups that have contributed to the curation efforts by selecting their names from the classification contributors pull down on the left. And also if one group curated a classification, but another did the final approval, the final approver can also be recognized under the classification approval pull down on the right. Upon saving the approved classification, it's permanently saved in the GCI as like a snapshot of data. As you can see, there's a link provided to the approved summary. Here you can go back at any time and link to the evidence summary that was reviewed and approved. And this will always be available to view. The final step, of course, is making your newly approved classification public on the ClinGen website. Upon approval, the option to publish appears. And when you click the publish button, the classification now has a published status in the GCI. In fact, a link appears that you can use to go view your newly published classification immediately. And a reminder of why due care and attention on that evidence summary text is important is that, as you can see here, on a real published gene validity classification summary, shown here on the right-hand side, the summary that was provided when the classification was saved in the GCI appears prominently at the top of the record. Below that, you see a table, a representation similar to the classification matrix in the GCI. One of the things our team and the website team are currently working on is uploading a more comprehensive summary of data, which is similar to the full evidence summary table I showed you a moment ago that GCEPs use as part of the expert review process. As of today, there are 1,369 total gene disease validity records on the ClinGen website from 1,106 unique genes that have been submitted by 39 different expert panels. I encourage you to go browse these curations, and for each one you can click on the link to see the classification summary that was approved. Now we have several new features under consideration for the GCI. As I said a moment ago, our intention is to show the full suite of evidence that was assessed as part of a gene disease classification on the ClinGen website. And this new feature actually is going to be delivered very soon. The website team are working on that now. In addition to this, we're in discussions at the moment with the gene tracker team led by Courtney Saxon at UNC to integrate the gene tracker with the GCI. The GCI already shares real-time updates for the gene tracker, but our plan is to provide users with a seamless curation experience as they traverse these two tools, which would include the ability to initiate a new gene disease record from within the gene tracker. We're also working on an API for the GCI so that ClinGen GCEPs can access and export all of their own data. And we're currently specifying what complex search issues users would like to be able to do within the GCI itself in order to find specific sets of records. And in doing so, allow the GCEPs and the Gene Curation Working Group as well to examine the curations within the GCI, which in turn will feed back to improve guidance on how to do gene curation. We're also looking at the ability to tag entries with labels that will help track the curation workflow within GCEPs and also provide automatic alerts within GCI records. So for instance, notifying GCEPs if the disease term has changed or it's time to do recuration of a gene disease pair. And actually, I didn't write it on here, but we're actually also looking at providing comprehensive audit trails, similar to the ones that are about to come out in the VCI. We're looking into that, so allow users to search on the curations history of each GCI record. A big thank you to all the GCI VCI team who all work to keep both VCI and GCI running and to keep updating these interfaces based on our user requests. I can honestly say it's a delight to work with all of them. And a big thank you to the gene curation stakeholder proxies, Erin, Courtney, Marina, Natasha, and G, who provide many of the specifications to turn those user requests into actionable items. Finally, if you require any help or if you have any questions about GCI, then please reach out to it. It's very memorable. It's gci@clinicalgenome.org. If you've got specific questions about, or if you want to get involved in gene curation, then that's genecuration@clinicalgenome.org. And if you have questions about ClinGen as a whole, it's clingen@clinicalgenome.org. And again, we also have lots of help information for you to read about the GCI in the header, all down in the header. Especially you can find in the header, there's a link to a help document, which actually runs through a lot of the things I've run through today, but a little bit more detail about how to use the curation workflow in the GCI. So that's me done, Jenny. So I'm ready for questions if there are any.
I don't see any questions in the chat. Does anyone have any questions for Matt? I guess one thing I was going to mention was the ability to modify a classification as well. I don't know if you want to talk about that, Matt. The reason I'm mentioning this is it just actually came up for one of the GCEPs I work in, and so it's a nice feature, and that information from that field will actually show up on the website as well. So if, for example, the points from your curation are kind of in between classifications or maybe just one direction or the other, and your experts feel like they want to change the classification compared with what is in the GCI, you can actually change that, but you must give a reason for it. And there's a box there in the approval field where you can do that. And as I said, that information does show up on the website. So just making sure that anything you put in there is you're okay with having that viewable by the public.
So when you talk about modifying, so you're talking about that really modifying the actual classification at the point when you go to approve.
Right.
Yeah, I think that might be actually slightly more like, so one of the conversations we've had recently with gene proxies was more if they get to the point of approval and they realize they've made a small error that doesn't affect the classification, but like something in the evidence summary or something like that. And that's something that's being specified at the moment, like, making small like error corrections and things that don't really affect the overall classification, but you don't want to have to go back and like re-provision it, reapprove it, just because you've made like some small typos somewhere in it. But I don't know about changing the actual classification at that stage. I think in that case, you should probably go back and actually put the information into the GCI, like, or else we'd end up with the situation we've got, like your snapshot that you're sending out has different information in it than the rest of the GCI itself. Although I guess we could look into if there are possibilities of ways of doing that. I don't know, it does sound slightly problematic.
Yeah.
Which system are you doing that in at the moment, where you do a modification right at the end?
Which system? So just, I guess I'm thinking for a situation, for example, where you're unable to add particular evidence. So just to let you know what the example was, in our retina JSEP, actually, there was a curation done and the points got to high moderate. But one of the experts in the group was aware of another case in a database that he's involved with. And they are actually planning to write up this case now, which is really cool because it will get that information actually out into the public. But until that point, we can't really add that evidence because it's not in a PMID. So we just simply at the end just said this is going up to definitive, and then we filled out the information in the modified classification box. And just said, we're aware of this other case, publication is being written up, but the experts want to make this definitive rather than moderate.
Yeah, and I think that kind of thing does happen at the moment. So, but as you say, what happens is at the moment is that people would manually change that classification and then write a reason and why they've changed it, and then maybe even put a note in their evidence summary as to why they did that modification.
Exactly. So just being very transparent about why that change was made. And like I said, I think it typically happens when points are, very borderline, and experts maybe feel like, well, it didn't quite make it to definitive, but we feel like it should be, or it did make it to moderate, but yeah, we're a little iffy. And they just, I think it just sort of points to the fact that the points are kind of a guide, but the experts can then look at the information overall and then decide what their final classification is, and just making sure that if they change it, that it's written in that modification box and that will go to the website.
I have a quick question for you, actually, which is just like in your role, because you were like a gene curator and you're involved in gene curation still. If folks on this call are really interested in getting involved in gene curation, how would you recommend they do that? What's the best avenue for people?
Yeah, that's a great question, Matt. So if you're interested in getting involved in gene curation, I would probably fill out the volunteer survey. Let me see if I can find that link, because that will allow you to say that you're interested. I'm just finding that right now because then I can put it in the chat. And then you would be assigned to a gene curation expert panel, and you would go through all of the training. Trying to find it on our website. And in the survey, you can also, here we go. You can also state if there's a particular disease area that you're interested in working in as well. So I'm going to put this in the chat. Okay. I think that's probably the best thing to do. But if anyone has any questions or anything, you're welcome to email me as well. Put my email in here too.
Well, thank you, Jenny, for your excellent moderator skills.
Oh, well, thank you all for these great presentations. It's just, yeah, I always think just amazing to see the amount of work that goes into these interfaces. Informatics is really, really great, and always fabulous to work with everybody. So it's great. I think we're at the end of the session now. So thank you so much to everyone for attending. So thanks so much, everybody.
Thanks again, Jenny. Thanks, everyone.
Thanks, everyone.